Hello everyone! Today me, Layla, Selena, Alexandra and Adamas will be talking about improving enzymatic degradation of plastics, an extremely important topic in today's environmental crisis. Plastics are so desirable because they are lightweight and chemically inert, making them durable and extremely cheap to produce. It is well known by everyone that plastic pollution is posing a giant threat to our natural environment. Each year, 359 million tons of plastic are produced, and 150 to 200 of these build up in the environment. Plastic's high resistance towards degradation has resulted in accumulation in the natural environment, meaning plastic particles exist in all ecosystems on Earth and can even enter the food chain. Current recycling efforts remain ineffective due to the amount of energy they require, as well as the fact that they often end up producing other toxic substances, resulting in secondary pollution. For these reasons, it is essential that we employ safer and more environmentally friendly methods of plastic degradation, by employing enzymes that are optimized for penetrating different plastic materials and depolymerizing them. Our study will focus primarily on the plastic PET, one of the most used plastics today. PET is an aromatic polyester which is spun into fibers and solidified into plastic. Strong and non-hydrolyzable covalent bonds between the PET monomers is what makes it so difficult to break down. There have been various discoveries of enzymes capable of degrading plastics, yet most are not able to be used at the industrial level. These enzymes can be divided into PET surface modifying enzymes and PET hydrolases. PET surface modifying enzymes have the ability to hydrolyze ester linkages on the surface of PET into polar hydroxyl and carboxylic groups. However, due to them only being able to degrade the surface of PET, this category of enzymes alone is not sufficient for complete PET degradation. PET hydrolases, on the other end, are able to degrade at least 10% of PET and perform hydrolysis. In Figure 4, the general mechanisms of action of the two types of PET degrading enzymes can be seen on a PET film. In 2016, a new bacteria capable of degrading PET at ambient temperatures was discovered. It was able to use PET film as a source of carbon and catabolize it for energy. They were then able to isolate this bacteria, known as Ideolena sakiensis 201F6. This degradation was thanks to two enzymes known as PETase and MHETase. PETase is able to degrade PET into MHET, which is then metabolized by MHETase. The products of this reaction are the environmentally friendly products TPA and EG. This process can be seen in Figure 5. Despite these exciting prospects, there are some limitations to these enzymes, their catalytic efficiency and their stability. While they are heat resistant and are able to degrade PET in more ambient temperatures, they degrade PET at a lower rate than other enzymes. As well as this, PETase activity tends to decrease over time. MHETase activity is inhibited by TPA, its product. Therefore, our aim is to suggest methods in which PET degradation can be improved, using a review of previous methods and experiments and our own interpretations. As for our methodology, our research is divided into two parts. First, the importance of the topic of PET degradation and the different enzymes involved are introduced. Then, the limitations of the two enzymes MHETase and PETase are discussed, and methods to improve them are presented. Information was retrieved from scientific papers found through the different search engines you can see on the slide.
Let's start with PETase. As mentioned, PETase degrades PET into mostly MHET with trace amounts of BHET and TPA.
Identifying the structure of the enzyme is crucial in order to improve its function. Studies done by Han et al. Provided great insight into the structural features of PETase. It was found that the enzyme consisted of 272 residues, the most important feature of the enzyme being the catalytic triad, which is responsible for the hydrolytic action against PET. The residues that take part of the catalytic triad are serine, aspartic acid, and histidine. Of course, there are also important features other than this catalytic triad that may contribute to substrate binding, its thermal stability, as well as enzymatic activity, that are found away from the substrate binding groove.
In this provided animation, you can see the structure and shape of PETase, with its alpha helices and beta sheets. Also, take note of the location of the catalytic triad, where hydrolysis of PET takes place.
Previous studies have revealed the advantages PETase holds over other existing PET hydrolases. Despite these promising characteristics, PET is not without its downfalls. PETases, as mentioned previously, are naturally secreted by I. sakaiensis, which are bacteria that thrive under mild conditions. It is because of this that the enzyme itself can only survive mild conditions, thus has low thermal stability. This low thermal stability means that the enzyme cannot survive the temperature fluctuations that may occur in the natural environment. The lack of survival would render it insufficient for practical use. Furthermore, other studies have also indicated that the enzyme has a slow rate of degradation. This would lead to a lack of significant results if used in the natural environment.
To improve upon the enzymatic activity of PTase, Ma et al. Took inspiration on previous studies where the enzymatic activity of cutinase was improved through mutagenesis. They investigated the change in enzymatic activity when several hotspot amino acids around the substrate binding group were altered to different amino acids. The study suggested that different interactions caused by the varying amino acid side chains impacted the enzyme to substrate interaction. During this investigation, it was discovered that a change from isoleucine to phenylalanine at residue 179 induced the greatest decrease in Km value and increase in enzymatic activity. To improve upon the thermal stability of PTase, Sun et al. Compared the enzyme to the more thermally stable cutinase. This comparison led to the discovery of a hydrogen bond found in the cutinase that was absent in the PTase. It was hypothesized that this could be the reason for the difference in their thermal stabilities. To test this theory, Sun et al. Also performed enzyme modification through mutagenesis to alter residues to allow for hydrogen bond formation. One of his variants was discovered to have formed water-mediated hydrogen bonding that allowed for not only an increase in thermal stability, but also an increase in enzymatic activity. Additionally, this variant was found to be able to survive 9 more days than the wild-type PTase in temperatures of 30 degrees Celsius.
Another important enzyme beside PETase is MHETase. MHETase degrades MHET, which is a product of PETase, to produce environmentally friendly terephthalic acid, or TPA, and ethylene glycol, or EG. Without MHETase, PTase would not be able to completely degrade PET. However, less is known about MHETase because most of the research has been focused on PETase, which is the rate-limiting component of PET degradation. The understanding of the enzyme MHETase has increased greatly thanks to more recent research. MHETase differs from PETase in that it's much larger. It has 611 residues. MHETase belongs to the tannase family, but is also very similar to furyl esterases. MHETase contains a lit domain, a catalytic triad, and both alpha and beta sheets. Both the lit domain of MHETase, which shields the active site, and a stabilizing calcium ion binding site are what makes MHETase different from other enzymes. The lit domain is associated with the regulation of catalytic activity and substrate specificity of the enzyme. The catalytic domain of MHETase has an alpha-beta hydrolase fold that is common in serine hydrosylases and contains a catalytic triad composed of serine, aspartate, and histidine. The lit domain also is a monomeric structure that lacks disulfide bridges, which consequently lowers the thermal stability of the enzyme. The calcium ion binding site is thought to induce conformational changes in the structure of the enzyme and causes an increase in substrate affinity as well as an increase in catalytic activity. It is composed of beta sheets, alpha helices, and a catalytic triad of histidine, aspartic acid, and serine. The calcium ion binding site is present to increase substrate affinity and catalytic activity. A problem with MHETase is that firstly, it is inhibited by its own product, TPA, when the concentrations of TPA are high, which means that TPA levels always need to be kept low for effective PET degradation. Another issue is the low thermal stability, which causes the enzyme to lose some of its activity if the temperature changes. In order to improve thermal stability, disulfide bridge formation can be induced through the introduction of cysteine residues. The method has worked for increasing thermal stability in furyl esterases, which, as I've already mentioned, are very similar to MHETase. Product inhibition can be decreased by altering the lit domain to change substrate affinity and specificity. For instance, introducing residues of negatively charged side chains have been found to increase thermal stability. To prevent turnover rate from decreasing, phenylalanine at position 495 and glutamate at position 226 should be conserved because they play an important part in substrate hydrolysis, although their mechanisms are unclear. In an industrial setting where substrate concentrations are high, enzyme activity could be increased by substituting tryptophan at position 397 with alanine. The substitution has a negative effect on substrate affinity, which could be overcome when the concentration of the substrate is kept high. In a natural setting, the conditions would not allow the low substrate affinity to be overcome, and the activity of the enzyme would decrease.
It is important to consider how PETase and MHETase can be improved together, since both are needed to fully degrade PET. PET degradation is best enhanced when both enzymes are present, which suggests that linking the two enzymes could increase the overall degradation. A longer amino acid residue linker has been found to enhance degradation better than a shorter linker. All the research conducted on ways to improve both PETase and MHETase involved inducing mutations to observe the effect on enzymatic activity. For both PETase and MHETase, areas of improvement included increasing thermal stability and improving its catalytic efficiency through base changes that resulted in amino acid alterations. We will now use all the information gathered to suggest a potential superior variance. Using previous studies, it is recommended that the most suitable mutation to improve the catalytic efficiency of PETase is to mutate isoleucine at site 179 into phenylalanine, which may improve catalytic efficiency of the enzyme because it resulted in the greatest kcat value compared to the other mutant variants produced in the study. In order to improve thermal stability of PETase, it is recommended to induce mutations at sites 121 from serine to glutamate and site 186 from aspartate to histidine, as this has been shown to form an appropriate intramolecular water-mediated hydrogen bond that increased thermal stability. A recommended alteration of MHETase to improve its thermal stability is to introduce a cysteine residue within the lid domain to form disulfide bridges that have been shown to increase the thermal stability of other similar enzymes. This recommendation has not yet been attempted on MHETase, but its success in other enzymes makes it a very promising alteration. To improve the catalytic efficiency of MHETase, the most promising alteration for industrial purposes was a mutation in which a tryptophan residue at position 397 was substituted with an alanine residue. Our final suggestion is to link the two enzymes using an amino acid linker composed of 20 amino acids. Further research must be conducted into PET's varying crystallinity, which affects the organization of the macromolecular aggregates of PET. Due to the varying levels of crystallinity, it is important the enzymes are able to bind successfully and catabolize them. An area for future research could be altering the residues around the active site to yield different sized binding sites in order to accommodate the different polymers. Another area for possible research is the secretion methods of the enzymes and how to improve them. Given the importance of enzyme secretion, methods to expedite the secretion process would allow for a greater volume of PET to be degraded. Attempts have already been made to improve this using model organisms. The importance of developing improved secretion methods is vital to tackle the large volume of plastic accumulating in the world today. Developing ways of degrading PET is crucial for protecting the environment from large amounts of plastic that is constantly accumulating. This is why I-Sachiensis, along with PETase and MHETase, are exciting discoveries that can provide a more sustainable way of breaking down PET. To optimize these two enzymes, 3 mutations are recommended, as well as linking the two enzymes together. On the next 4 slides, our list of references can be seen. Thank you for listening.